Question number 1. Genetic information flow from DNA to DNA, DNA to RNA, RNA to cellular proteins, DNA to cellular proteins. Correct answer is option B, DNA to RNA. Question 2. Genetic code is collection of codon, collection of amino acid, collection of purine base, collection of pyrimidine base. Correct answer is option A, collection of codon. Question 3. Degeneracy of genetic code implies that codon do not code for specific amino acid. Multiple codon must decode the same amino acid. No anticodon on tRNA molecules. Specific codon decodes many amino acids. Correct answer is option B. Multiple codon must decode the same amino acid. Question 4. Genetic code is overlapping, non-overlapping, not universal, ambiguous. Correct answer is option A. Correct answer is option B. Genetic code is non-overlapping. Question 5. mRNA is a complementary to the nucleotide sequence of correct coding strand, ribosomal RNA, tRNA, template strand. Correct answer is option A. Coding strand. Question 6. In DNA replication, the enzyme required in the first step is DNA-directed polymerase, unwinding proteins, DNA polymerase, DNA ligase. Correct answer is option B, unwinding protein. Unwinding proteins कौन सा हेलिकेज. Question 7. The smallest unit of DNA capable of coding for the synthesis of polypeptide is operon, repressor gene, cistron, replicon. Correct answer is option C, cistron. Question 8. Termination of the synthesis of RNA molecule is signaled by a sequence in the template strand of the DNA molecule. A signal that is recognized by a terminator protein is rho factor, sigma factor, delta factor, epsilon factor. Correct answer is option A, rho factor. Question 9. After termination of the synthesis of RNA molecule, the core enzyme separates from the DNA template. The core enzyme then recognizes a promoter at which the synthesis of new RNA molecule commences with the assistance of rho factor, sigma factor, beta factor, sigma factor. Correct answer is option D, sigma factor. Question 10. In the process of transcription in bacterial cell, initiation requires rho protein. RNA polymerase incorporate methylated bases in correct sequence. Both the sigma unit and core enzyme of RNA polymerase are required for accurate promoter site binding. Primase is necessary for initiation. Correct answer is option C. Both the sigma unit and core enzyme of RNA polymerase are required for accurate promoter site binding. Question 11. The correct statement concerning RNA and DNA polymerase is RNA polymerase use nucleoside diphosphate. RNA polymers require primers and add base at the 5' end of the growing polynucleotide chain. DNA polymerase can add nucleotides at both ends of the chain. All RNA and DNA polymerase can add nucleotides only at the 3' end of the growing polynucleotide chain. Correct answer is option D. All RNA and DNA polymerase can add nucleotides only at the 3' end of the growing polynucleotide chain. Question 12. The eukaryotic nuclear chromosomal DNA is a linear and unbranched molecule, is not associated with specific membranes organelle. It is not replicated semi-conservatively. It's about the same size as each prokaryotic chromosome. Correct answer is option A, is a linear and unbranched molecule. Question 30. The function of repressor protein in an operon system is to prevent synthesis by binding to the ribosome. a specific region of the operon preventing transcription of structural gene. The RNA polymerase, a specific region of the mRNA preventing translation to protein. Correct answer is option B, a specific region of the operon preventing transcription of structural gene. Question 40. All Pribnobox are variant of the sequence. Correct answer is option A, 5-2 TATAAT. Question 50. 5-day terminus of mRNA molecule is capped with guanosine triphosphate, 7-methyl guanosine triphosphate, adenosine triphosphate, adenosine diphosphate. Correct answer is option B, 7-methyl guanosine triphosphate. 16. The first codon to be translated on mRNA is AUG, GGU, GGA, AAA. Correct answer is option B, AUG. 17. AUG, the only identified codon for methionine, is important as a releasing factor for peptide chain, a chain terminating codon, recognition site on tRNA, a chain initiating codon. Correct answer is option D, a chain initiating codon. 80. In biosynthesis of protein, the chain terminating codons are. Correct answer is option A, UAA, UAG and UGA. 90. The formation of initiation complex during protein synthesis require factor. Correct answer is option A, prolongation factor 3. Question number 20. The amino terminal of all polypeptide chain at the time of synthesis in E. coli is tagged to amino acid residue methionine, serine, N-formyl methionine, N-formyl serine. Correct answer is option C, N-formyl methionine. 21. Initiation of protein synthesis begins with binding of 40S ribosomal unit on mRNA, 60S ribosomal unit, charging of tRNA with specific amino acid, attachment of amino acid tRNA on mRNA. Correct answer is option A, 40S ribosomal unit on mRNA. 22. Initiation of protein synthesis require. Correct answer is option D, GTP. 23. The enzyme amino acid tRNA synthesis is involved in dissociation of discharged tRNA from ATS ribosome, charging of tRNA with specific amino acid, termination of protein synthesis, nucleophilic attack on esterified carboxyl group of peptidyl tRNA. Correct answer is option B, charging of tRNA with specific amino acid. 24. In the process of activation of amino acid for protein synthesis, the number of high energy phosphate bond equivalent utilized is. Correct answer is option C, 2. Translation result. Question 25. Translation result in a product known as. Correct answer is option A, protein. 26. In the process of elongation of chain binding of amino acid tRNA to A site requires a proper codon recognition GTP, elongation factor 2 GDP. Correct answer is option A, a proper codon recognition. 27. A newly entering amino acid tRNA into A site require elongation factor 2, ribosomal RNA, mRNA elongation factor 1. Correct answer is option D, elongation factor 1. 28. The alpha amino group of new amino acid tRNA in the A site carries out a nucleophilic attack on the esterified carboxylic group of the peptidyl tRNA occupying the P site. The reaction is catalyzed by DNA polymerase, RNA polymerase, peptidyl transferase, DNA lipase. Correct answer is option C, peptidyl transferase. 29. The nucleophilic attack on the esterified carboxyl group of the peptidyl tRNA occupying the P site and the alpha amino group of the new amino acid tRNA. The number of ATP required by the amino acid on the charged tRNA is Answer is option A 0. Question number 30. Translocation of the newly formed peptidyl tRNA at the A site into MTP site involves. Correct answer is option A, elongation factor 2 and GTP.